we'll start out with, I'm sure the clicker questions are ridiculously easy today, right? By definition. Let's see. Virus with the largest virion known to date is from the genus. Actually, this should have been with infecting cells with this. Fuselloviruses.
Yeah, it almost got memorized. I mean, these parasites.
My virion is bigger than your virion.
Darn Frank.
Bah oui.
Is there any idea how SSV1 gets around?
10. Three. And no one likes Ebola, although actually they got really long virions, but they're not in terms of volume. And in fact, even the length of those virions is way less than Pandora virus, which is less than Pithovirus. So Pithovirus is in fact the largest of those known to date. So, but again, these virion, yes. Genome size, Pandora is larger, but the virion is larger. Yeah, but the actual virion, the Pithovirion, is larger than the Pandora one. So maybe I was incorrect in the notes, but this is in fact the largest. So I can go back and check now that you've made me to go back and check these things. Well, numbers, who cares about numbers? It's just that's what Stedman always likes to have on his multiple choice questions, right? Which is the reminder that when is the exam?
Tuesday, 8 a.m.
8 a.m. Tuesday next week. Be here. Don't be late. Okay, so Archaeal viruses. Yes, the coolest viruses that we're going to talk about all term. And yes, I have framed pictures of them in my office. Gives you a bit of an idea why. These are the things that we work with in my lab, although we only work with very few of these Archaeal viruses. There are a whole bunch of them. It's kind of like talking about all the viruses of eukarya in one lecture, which is really kind of ridiculous. So, or viruses of bacteria for that matter as well. So unfortunately, it's going to be a very sort of high-level overview, and when we get another 50 faculty members, I can actually teach a real course on the viruses of Archaea. Almost all of the viruses so far found in Archaea and clearly shown to be archaeal viruses are double-stranded DNA viruses, with one exception that we will talk about. In general, and again, this is even more ridiculously broad brush overview than we've normally done so far, the viruses which infect the Archaea are bacterial-like in terms of their viruses, so they're a lot like the bacteriophage. On the other hand, those that infect the Archaea, which is the other major, at least used to be the other major branch of Archaea. Anyone go to the Baker seminar a couple of weeks ago? Biology, yeah, so there's a ridiculous number of archaeal branches now, but we thought were the two major archaeal branches, Archaea and Archaea. The Archaea, these are the really bizarre, crazy, wacko viruses. Unique morphology, unique genomes, all kinds of really fascinating aspects about these viruses that we'll talk about in a little bit more detail. Again, most of the viruses of Archaea are phage-like in general, but that's probably because a lot of people were looking for phage or phage-like particles when they were looking at archaeal viruses. And it was only considerably later when particularly the work of Wolfram Zillig, my postdoctoral mentor, and some of his colleagues really started to focus in on some of the other viruses, including our friends, the Fuselloviruses up here in the corner, which we're going to spend most of the time talking about because that's the stuff we do in my lab. But we'll also talk a little bit about some of these other virus families. And basically, these are like virus families like each of the chapters in our textbook has been. So each of these could have their own textbook chapter relative to each other here. Rudiviruses, we'll mostly spend our time talking about the Rudiviruses. These are the rod-shaped viruses because there's been some really fascinating work that's come out in the last couple of years to try and understand how these things are fitting together in terms of the virion. This is what 99.9% of the virologists think about when they think about the virosphere, i.e. where all of these viruses are. We've already talked about some of these guys. That would be the mimiviruses, some of the baculoviruses, pox viruses, the double-stranded DNA viruses, single-stranded DNA viruses, positive-strand RNA viruses, negative-strand RNA viruses. We haven't talked about the double-stranded RNA viruses, and then these reverse transcriptase viruses. And there's this little tiny segment right here on the Archaea. In my opinion, it should be about this big, because again, each of these guys represents one virus family. Each of the individual icons here represent virus families. So again, people have no idea about how cool these viruses are. Why study viruses of Archaea? Hopefully Archaea means something to you now. If anything, PSU is one of the strongest places in the world in terms of study of Archaea. So think yourself lucky to be here. Archaea are absolutely everywhere. Soil, oceans, freshwater, your gut, skin, etc. They're very similar to eukaryotes at the basic molecular biology level. And so that's one of the reasons that we study them, or at least try and get money to study them. But the thing that gets me really excited about working with these particular organisms is they love extreme environments. And in fact, this is where they're originally found. Boiling acid hot springs, basically. And everything I had learned about life, biochemistry, etc., says this is not very good for life, but they're teeming with life. And a lot of those are Archaea. There are a couple of interesting aspects that may have to do with how they can survive under those conditions. One is they have ether lipids rather than ester lipids, which is potentially stabilizing process, so allows them to survive in some of these environments. And it may well be that they're able to continue to survive in some of these environments when the nasty bacteria and eukarya, which have ester lipids, are surviving in other environments. And so it may well be that these are refugia where these organisms have been able to still manage to outcompete everybody else. We're mostly going to talk about Sulfolobus here. In this particular place in the old big tree of life based on small subunit RNA sequences. And for reference, you are here, as also 90% of the viruses that we've talked about so far this term are here, or viruses which are infecting E. coli up here in bacteria. One other thing about this tree that tells you, since we're talking about 90% here and here, and then just this lecture today, means there's a heck of a lot of other viruses out there that we know nothing about, and those potentially getting way back to lecture 1, all those little dots. All those little dots that we had. Look at oceans, look at soils, look at any environment, lots of dots. And those are viruses infecting things that we have yet to characterize anything from. But at least in viruses of Archaea, we have a couple of really fascinating things that have got people working on them for 20-some years, which is how long as I've been working on them. So most importantly here, these are the virions that are very, very different in their structure. We'll ignore these guys for the time being. These are the boring ones that actually look a lot like bacteriophage. We already talked about STIV, the Sulfolobus turridic icosahedrovirus that I found in Yellowstone. We're going to spend a long time talking about these Fuselloviruses, these lemon-shaped particles. This is the three-dimensional image, about 1 million times life size here. But there are also some other really fascinating morphologies that haven't been seen anywhere else. And probably the most spectacular of these are the Acidianus bottle-shaped viruses that we'll talk about a little bit later on, with this sort of ring of almost look like birthday candles at the top here. My daughter's 9th birthday was last week. And this kind of champagne bottle structure here. How the heck you put this together with regular subunits is a really completely open question. So let's look at the boring viruses of Archaea. These, again, were originally found because people were looking in the electron microscope for things that they thought looked like viruses, which would be your heads, your tails, the classic kind of thing. So one of the very first to be isolated was this one right here, Phi-H. So Phi-X174, Phi-H, Phi-H4, an extreme halophile, was found in some of the very first discovered archaebacteria, which live in saturating salt solutions. So this was one of those very first ones. Soon thereafter, methanogens, which are the very first discovered of the Archaea. They found viruses associated with them. This is Psi-M1. And then as people were now looking, and again, when I say people, mostly Wolfram Zillig and his colleagues, were looking for viruses of the Archaea in these high-temperature acidic environments. They found these really funky morphologies like we looked at on the last slide. Then people went back to some of these extreme saline conditions, some of the methanogen conditions, and found viruses there that looked a little bit like these bizarre, funky things that people had seen before. And so that led to the discovery of the His1 virus, which is back up here. This has a spindle shape, in fact, very similar to SSB1, and this is the model, obviously smaller, so much less important than SSB1, but having a really amazingly similar overall virion structure. Now, why something that's functioning at 5 molar salt or 8 degrees C and pH 3 should have such similar shapes? Any good guesses? Let me know, because we really have no idea. And also, based on the STIV virus that I had discovered, people soon thereafter discovered this virus, icosahedral 1, which looks a lot like STIV, particularly in cryo, also has nice projections of the 5-fold axes of symmetry. So, yeah, most of them, again, particularly the earlier ones that were found, the viruses that infecting the Archaea, were those which had structures similar to bacteriophage, but more recently people have found more and more of some of these other ones. Extreme thermophiles, turns out that there are Archaea that also function at very, very high temperatures. Anne-Louise Reisenbach, how many of you know Dr. Reisenbach? Pokes around deep-sea hydrothermal vents, finds really fascinating creatures down there. A lot of these extreme thermophiles also have viruses that are associated with them. They're Archaea, not Archaea. Some of those look a little bit like our SSBs, the Fuselloviruses down here as well. But one of the best studied of these viruses of the Archaea is a virus called Phi-H or Phi-H1. Seem to be basically interchangeable. And the main thing here is that it looks practically identical to a bacteriophage. This is bacteriophage B2. This is Phi-H. And so in terms of their morphology, practically identical to each other. Curiously enough, however, when you look at the genome and how the genome is regulated of Phi-H relative to P2 that we really haven't talked about here, it looks a lot like bacteriophage lambda. So it's got divergent transcripts, it integrates into the genome, it really seems to function in a very similar way. It's a linear, but circularizes once it comes inside the cell. So it's very much like lambda. So that's all we're going to talk about in terms of those Archaeal viruses, because they're boring. No, it's because there's actually a lot less known about them. We don't work with them as much. Switch gears, talk a little bit about the viruses of Sulfolobus. They're present in these high temperature, low pH environments. This particular hot spring in Yellowstone National Park was 85 degrees Celsius and pH 3 at the time we were there. With HDMI, it looks a little closer to the right color. Really does kind of look like tomato soup. And to give you an idea on what skinflints us professors are, like this guy here who has a little less gray hair in this picture than he does now, he still has the same fleece. And you can see the date down at the bottom there. So gives you a bit of an idea what the approach is for a lot of these things. How do you collect samples from these environments? Basically with a really long pole, and get yourself as far away from it as you possibly can before you collect a sample. The STIV virus was found in a sample about 100 yards away from where this picture was taken. So not, in fact, in that spring. Yeah?
Do you still have those virions in your lab?
The virions?
Yeah.
That we have collected from there. So we have the cells that we've collected them from, and then we can grow the virions from those infected cells.
So you can culture them?
We can culture them, yes. So, in fact, that's what we do in the recombinant DNA techniques lab. We culture them, although not the STIV one. We're culturing lots of those that are these SSV-like viruses. So, in the interest of really easy clicker questions, let's do another one, hopefully. Virions of viruses which infect which of the following have the most known morphological diversity? Archaea, Archaea, E. coli, primates, or amoeba? Blue. I was really tempted.
In Africa, it's like yellow and green, sulfur, bubbling. It's in the
So there's some hot springs in the Rift Valley. Most of those are alkaline springs. We haven't sampled there, but some of our colleagues have.
They always show pictures of just such a alien-looking place. You see all this sulfur everywhere.
Yeah. No, we've got some pretty alien-looking environments that we sampled into.
Are you guys carrying gas monitors when you're around there?
There's this gas monitor,
Yeah.
which when you can't smell the hydrogen sulfide anymore, it means it's really bad. We don't. Some people do. But most of the places we do, there's pretty good circulation, so it's not such a big deal. Yay, yes, it's the Archaea, because they're clearly the most important and interesting ones. Just think, only 2 more clicker questions for the whole term. Sigh. Which does remind me, I've been trying to upload them. This computer, again, has been having some issues with connecting to the world. I have all of the clicker questions. They will be posted eventually. So let's talk about the Fuselloviruses. These are, of course, the most interesting, exciting ones. It's mostly what we were working on. This was actually one of my very first electron micrographs that I ever took, which I was quite happy about. Each of these are the individual virions. This is a piece of cell debris that all of them are attached to here, probably through these little tail structures. This was the very first of the Archaeal viruses to be discovered and actually studied in any detail. Was originally found because it had this really beautiful structure, but also people were originally interested in it just because of the genome, because a lot like lambda, again, everything you know about molecular biology comes from the study of lambda. The thought was everything you need to know about the molecular biology of Archaea comes from the study of SSV, which to some extent is true. A lot of what we know about Archaeal transcription, some of what we know about Archaeal translation, a little bit of what we know about Archaeal replication comes, in fact, from the study of SSV, but particularly the transcriptional aspects of things. The genome, which is packaged on the inside of each of these virions, is a double-stranded circular genome that's positively supercoiled. So why is positive supercoiling such a big deal? Well, what positive supercoiling does is it overwinds the DNA and basically protects it from being thermally denatured, i.e. being pulled apart at high temperatures. Well, that's exactly what you want to have if you're trying to function at 80 plus degrees Celsius. And so having that positively supercoiled DNA is a really novel form of packaging of your DNA. Now, how that happens is a still very open question, and what it does about getting packaged inside the virion is, again, a very open question. The genome of SSV integrates specifically into the host genome, a lot like what happens with Lambda, and there is, in fact, a integrase gene that is important for doing that. There's a UV induction, so you shine UV light on an infected culture, you get a big burst of production of virus. These two things should sound a lot like Lambda, exactly, specific integration and UV induction. And this particular virus was found in southern Japan, island of Kyushu, a place called Beppu, Sulfolobus shibata, and we'll get back to that a little bit later on. This positively supercoiled DNA genome is relatively small. It's only about 15,500 base pairs in size, but very, very little was known about it when I started working on it, which admittedly was 20 years ago. We now know a little bit more after 20 years of work on it, but not that much more. And so we'll talk about some of that here. So we knew at that point, again, before I started working on it, that these three genes right here, VP1, VP3, and VP2, were the major viral structural proteins. More recently, actually last year, it was shown that this gene over here encodes another protein that you find in virions, probably this tail structure. I mentioned before, the viral genome is integrated into the host genome. That's with this viral integrase protein right here. What's interesting about this viral integrase protein is that the integration site, the attachment site, is actually in the integrase gene. That's what this bar is here, the at-P site. And what that means is when you integrate your genome, you take the circle, you break it open and pop it into the host genome, you've actually broken up the integrase gene in that process and presumably inactivated it. So the integration process seems to be a one-way street. Once you've gone in, you're in. And unless you have that protein around, you're never going to be able to transcribe and make more of that protein. Yeah, Patrick?
Is the break site in the integrase gene that black bar?
Yeah, exactly. That black bar right there is where the break point is. So for the rest of it, we didn't really have a clue. So lots of different ways to approach trying to figure this out, partly farming it out to our recombinant DNA techniques lab, but partly coming up with some new techniques in terms of doing mutagenesis on some of these viruses. The genetic approach, I did my PhD working on the genetics of Salmonella typhimurium and how genes were being turned on and turned off, but using a very genetic approach. So I wanted to use a genetic approach to studying some of these viruses. So Adam Kalor, a PhD student of mine, also graduated 10 years ago, kind of frighteningly, came up with this technique we call long universe PCR, which is a pretty straightforward process. Basically, instead of with standard PCR, we have primers that face each other, and you amplify between the two of those primers. You have primers that face away from each other, and as long as you have a circular genome, you can amplify from there and amplify everything except what's between those two primers. So sort of the inverse, what you think about PCR, between the two primers. And then what you can do is hook the two of these two together, and in the process, delete whatever it is between those two particular primers. Adam did this for the viral integrase gene because we thought it was something we knew something about. We figured that would kill the virus. Of course, it didn't. That's what usually happens when you do experiments. You have to at least do them to figure out what's going on there. And then more recently, Eric Iverson, graduate student who finished up last year, has basically been continuing to do this through the rest of the viral genome. Now, why has he been doing this? I mentioned right at the beginning we knew something, or at least we thought we knew something about what these three genes did, and we thought we knew something about what this gene did. All the rest of the genes in the genome, we had no clue what the heck they were doing. And actually, we still don't know, for the most part, what most of them are doing. So one of the things that we wanted to do was just to systematically go through and see if we could get rid of some of these genes and still have a functional virus. And that overview of, I don't know, So Eric spent about 6 years doing his PhD, and I'm going to give him 2 slides in about 5 minutes. So what he did was he went through and did a whole bunch of deletions in the genome, basically using that exact technique, that same long inverse PCR technique. And what he found, let's just focus up here at the top. You remember VP1, VP3, and VP2, those virus structural proteins? You can actually get rid of 2 of them, and the virus still seems to be perfectly happy. Okay, wait a minute. So we can get rid of those 2, and then we can get rid of the integrase genes. So the 4 genes that we thought we knew something about, we can get rid of 3 of them, and the virus is still happy. Okay. We did, however, find a couple of genes, these ones that are here in red, that were in fact, seem to be important for virus function. This open reading frame B129, this one over here, and the VP1 protein encoding gene, that major structural protein. So those did, in fact, seem to be important for virus function. It took Adam Clore about 2 years to figure out how to do the first deletion. Eric got it down to about 6 weeks per deletion, but he figured that this was still taking a little bit too long, and he did actually want to get his degree. So he decided that we could do some more random mutagenesis, and that's all these arrows out here on the outside. These represent insertional mutants, some of which were made by Eric, some were made by David Goodman, who's the TA of some of you in the recombinant DNA techniques lab. Valerie Wong was a high school student in our lab, and then a number of these arrows around the outside have actually been contributed by people who've been taking the recombinant DNA techniques lab. And so the color of each of these arrows represents whether the virus can actually function with an insertion here, which would be a green arrow, these guys over here, or a red arrow that says the virus can't function when you have an insertion in this particular part of the genome. And if you're interested in more about this, come down to CLSB this afternoon and tomorrow and hear all the presentations and people in the recombinant DNA lab who will tell you about all the insertions which they've been making into the virus genome and how they've been able to interpret it. But one of the things which is really pretty amazing here is you can mess around with this genome a whole bunch and it still seems to be relatively happy. So that's one way to approach figuring out, or at least trying to start to figure out what some of these unknown genes are doing, or maybe even some of the known genes up here at the top. Another way to do this is to let nature tell you. And this is the fun part about studying these particular viruses. And that is, turns out the reason that they were able to find this virus relatively easily is that if you go to high temperature, low pH environments, you find viruses like these SSVs all over the place. You find them in Iceland, you find them in Kamchatka, you find them in Yellowstone. This is actually me again with slightly less gray hair. This is Wolfram Zillig, who was really the pioneer in the field, studying Iceland and talking about moon or alien landscapes. Iceland's a really good example for that. And I was very fortunate to be able to go to Kamchatka in far eastern Russia in 2000, right before there was a big landslide that wiped out this whole area. Give you an idea how remote this place is. It's only accessible by helicopter. Helicopters that look like they're in the last set of wars in Afghanistan. And there are more geysers there than there are in Yellowstone. Was discovered by Westerners in the 1940s. So really very isolated place. But high temperature, low pH springs, perfect kind of environments for Sulfolobus and the viruses which infect them. So if we look at number of samples from Iceland, we've sampled in Yellowstone. We've also sampled in Lassen Volcanic National Park in Northern California. The original SSV was from the southern isle of Japan. And then we have a few of these viruses from Kamchatka. Why bother? Why go through this whole process? Well, you can isolate viruses from these places, and then what you can do is sequence their genomes. And in sequencing their genomes, what you do is you find that some of the genes are present in every single one of these viruses, wherever they are, any different part of the world, are well conserved. They're probably going to be important for how the virus is functioning. And then you'll have other genes that are a lot less well conserved. And that's exactly what we found here. These are now, I think if I remember correctly, 10 different genomes. We're up to 18. The slide's always out of date now. This particular one down at the bottom, in fact, is from a Mexican hot strain. Sulfolobus, I think they call it Mexicanus, I forget. But if you look at all of these genes, you see that about half of them are present in every single one of the genomes. And then as you go further down this list, the white ones are only present in one of these different viruses. So a lot of these areas down here that are white, where we have no other viruses that have them, that's also where we're able to make all these kinds of mutations that we had before. Whereas on this side of the genome, where it looks as if all these genes are very well conserved, with a couple of interesting exceptions, here we can't make very many mutations and still have something which is functional. So as an overview, what we talk about in terms of SSB genetics, partially conserved. Again, about half the genome is well conserved. You find them all over the place, but not all genes are essential. We can mess with a whole bunch of them. Trevor, just a second. And then with these different genomes, one of the things that I didn't mention is if you look at all the viruses that you find from Iceland, they're actually more similar to each other than the viruses from Iceland are to Kamchatka, or the viruses from Japan are to the viruses from Yellowstone. And so there does seem to be a little bit of a geographic separation to each of these different viruses. And we have some ideas why that is that we may or may not have a chance to talk about later on. Yeah, Trevor.
So based on the image that you showed, it seems like it's very regional. Like the lower region are the ones that, they can be changed. And it's conserved throughout, that there's like an area along the genome that's more changeable than the other.
Yes, so that's exactly right. Well noticed that about the top half here is very well conserved. The bottom half much less so. And a couple of things that you may or may not have noticed here are these little T's in the middle. These represent transcripts. And so these are the messenger RNAs which are being made, which will then be translated into proteins. And if you look particularly in transcript 5, there's very little conservation and lots of places that we can make mutations in those. Back up here to here, lots of places you can make mutations in transcript number 5. Transcript number 6, a little bit of that. As soon as you get into transcripts 4, 7, and 8, 1 and 2 are weird. And transcript 9, you can make very few mutations in these.
So it looks like whatever around the integrase gene and then whatever's the C124 gene. Are there any flanking genes that would make sense as to why this is the changeable area?
There's not anything really obvious about why there would be one. Probably just that there's a core set of genes that are really well conserved, and then a non-core set of genes which are pretty variable. And people have seen this particularly in bacterial genomes, bacteriophage genomes as well. There's a set of what are called core genes and then a variable gene set. You also see this in bacterial genomes. You have a set of core genes and then a set of variable genes. Rarely, at least in the bacteria, they're all set together quite as easily, but that's one of the things you see. Yeah?
Doesn't the host vary from region to region as well?
Yes, so the hosts vary from region to region as well, which is a nice lead into host range, but I'll answer Patrick's question in just a second.
And so I was just saying, there would be variability in the genome of the virus, but couldn't that also be explained by the variability of the hosts?
Right, so we'll get to that in just a second, but yes, it's certainly possible. And we do see the same kind of geographical differentiation. So 2 Sulfolobus from Iceland are more similar to each other than a Sulfolobus from Iceland and a Sulfolobus from Yellowstone, for instance. Yeah?
The conservation slide, the TX
Yeah.
for C124, transcription X, is that because we don't know at what stage it's transcribed, or is it because we don't know if it's transcribed?
So why is TX called TX rather than having a nice number? The reasoning for that has to do with the history of the naming of all of those transcripts. And so first 9 transcripts were found, and so they were named 1 through 9. And then literally a decade and a half later, another transcript was found. And so what do you call it? You could do something like T7 and call it 0.1 or 0.5 or 3.5 or something like that, or just call it TX. So that was the reasoning behind that. Yeah, so great question about the hosts. And one of the questions that we immediately tried to answer, and we saw this differentiation in terms of virus genomes from different parts of the world, differentiation of hosts from different parts of the world. is there something about some coevolution that was happening between the two? Exactly what you'd expect is that you have viruses infecting Kamchatkan hosts are going to be adapted to each other. Viruses that are infecting Japanese hosts are going to be adapted to each other. And the great thing is we have them in the lab. And so we can do these kinds of mixing experiments in the lab and say, okay, does the virus from Yellowstone infect the Sulfolobus from Iceland, etc.? So that's the host range study. And the way that we do this is, again, very similar to what we've been doing in the recombinant DNA lab. It's kind of a plaque assay, but scaled down a little bit. Basically what we do is we take a culture of Sulfolobus, mix it together with this stuff called gelrite, because agar has problems at 80 degrees Celsius, like it's liquid. But gelrite does nicely. Put it onto a Petri plate again of gelrite, let it solidify, and then just literally spot on this plate with whatever Sulfolobus culture it is, various different viruses. And if they're functional, your virus will cause a zone of clearing on your plate. If they're not functional, or this is not a host for that particular virus, you'll see normal growth of the archaea on the plate. And so this is the work that Caleb Marceau, PSU graduate in biology, 2009, soon to have his PhD from Stanford, did. Basically took all kinds of different Sulfolobus and all kinds of different viruses and mixed them all together and said, okay, are they infecting, are they not infecting? And what he saw was actually really quite interesting, is that all of these Sulfolobus strains, so different Sulfolobus strains from Italy, can get infected by a whole bunch of different viruses, and this is color-coded for different countries. So SSV1 is from Japan, SSV2 and SSV3 are from Iceland, SSVL is from Lassen Volcanic National Park, SSVK1 is from Kamchatka. But the basic take-home message here is that there's no obvious virus from one place infecting a host from another place, or that hosts from one place are only susceptible to viruses or not susceptible to local viruses. And so it's really not clear what's going on. So getting back to your question about, aren't these differences due to the differences in the hosts? Yes, maybe, but there's no obvious way to pull them apart relative to each other. And that's one of the things that we're trying to do, is to figure out why, for instance, the Italians are great hosts. We kind of knew that already. And we're really good hosts as well, but we can also get infected really well, so that's not so good. The Japanese are a little more reticent in terms of allowing viruses to infect them, and the Russians are all over the place. So this is, again, a very open and interesting area to try and look at, because we don't know what the host range is. We don't know what the parameters are in terms of which host can be infected by what virus, etc. So that's all I wanted to talk about in terms of the Fuselloviruses. Again, we could spend all term talking about them. In fact, they did in the recombinant DNA class. They integrate lots and lots of genes that don't resemble anything else in the database, but lots of them are not required. We can make lots and lots of changes to the genome, and it still is functional. This is a big surprise. We didn't expect to be able to mess around with it. Most viral genomes, you can't beat all over and still have the virus be functional. Strangely enough, you can for this. Find them all over the place, and we're still trying to answer questions about the host range here. No obvious connection between the variability in the virus genomes and the variability in the host genomes. One of the obvious things that people immediately said, what about CRISPRs? Because the Sulfolobus have all of these CRISPR repeat structures. CRISPR is the microbial defense system, microbial immunity. There's no obvious connection between CRISPR matches and host ranges as well. So we thought that was a great answer, but turned out actually not to be the case. So last clicker question for today's lecture, I think. Maybe I put in another one, I can't remember. Hosts from which region of the world seem to have the least hospitality for SSVs? Italy, Iceland, Kamchatka, Japan, or the USA?
It's a weird little diagram. Yeah,
If
given
you're recording,
our
you can-
work in the lab, I kind of appreciate how much of a grind that must have been for him.
It took him a couple of years.
Yeah.
Yeah. The rest of today's lecture, depending on where we get to. Five. No such luck. No 100% today. So that it is, in fact, very strange that most of these Japanese isolates do not serve as hosts. So Japan is the correct answer here. I'm, in fact, going to Japan in September to try and figure this out and try and collect a few more samples and see quite why this is the case. One of the things which is also very strange, getting back to your question about host diversity, it looks like there's more host diversity in Japan than anywhere else in the world. Why that is the case is really not clear, again, at all. But yeah, the Italians in the U.S. are great hosts, in general. So I also like to go to Italy and collect there. A wonderful place to collect and really good food. But that's a different story. So to finish up today, which is, again, a horrible term for this, going to give some overview of a number of different virus families. And again, remember, this is like one chapter per slide as far as what we're going through here. So the Lipotrix viruses, this is a set of viruses, again, found in Sulfolobus samples from Iceland, where people were looking for something which caused the cells to be sick, looked under the electron microscope, saw what they could see, and to their amazement, they saw these ridiculously long particles. These are almost 2 microns in length. The poor Sulfolobus is only about a micron in diameter, and so these virions are twice as long as the actual host that they're infecting, which is really bizarre. But this is certainly clearly what's happening there. These also have these sort of nano-sized claws at the ends of their virions. There's the cartoon right here, which then close on pili, which seems to be the way that these actually are infecting the cell. And so these terminal structures. Now, how that mechanically actually works is a really fascinating question. Lots of nanotechnology people are trying to look at that as well. Above and beyond the interest in terms of the virion structure, these are double-stranded DNA genomes with telomere-like ends. And so they've got these repeated structures. Again, I think they're 5 or 6 nucleotide repeats. They repeat again and again, and also inverted terminal repeat structures. So very often, again, we've been talking a lot about inverted terminal repeats for linear DNA genomes. These guys have that very typical kind of structure. This was again discovered in Wolfram Zillig's lab while I was there. First the Sulfolobus islandicus filamentous virus, and then the Acidionus filamentous virus, all of which have very similar kinds of structures here. One thing that's not quite as obvious is these are very flexible rods, shown here more in the cartoon form down at the bottom. These are really pretty floppy relative to something like one of these spindle-shaped viruses, and when we talk about the Rudiviruses a little bit later on, those are very stiff rod-like structures. What appears to be happening is there's a helical packaging of this double-stranded DNA inside an envelope on the outside, and that envelope is the lipo, because it's a envelope. These are lipids on the outside of the particular virus. Probably the most amazing of the virion morphologies, as I mentioned before, is this bottle-shaped virus. originally isolated from Acidianus. This is a sample from Italy, in fact, just outside of Naples, a place called Pigerelli, which is where the original Sulfolobus sulfatericus is from, right next to a tennis hotel. But this particular sample here doesn't infect Sulfolobus, infects the very similar relative of Sulfolobus acidianus. Here's the virion again. It's got these projections at one end, these short, stiff filaments at one end, and then very much a plug with a bottle-like structure down here at the bottom. It looks as if the genome is wrapped around sort of a central core structure right here in the middle. Again, double-stranded DNA virus. Looks like it has protein-primed replication, rather like what you see with what viruses? Protein-primed replication? Okay, herpes is usually not protein-primed. That's RNA primed, but polio is protein-primed and adenoviruses. So in terms of the exam that's coming out. Yes, so protein-primed, terminal proteins. So the OH is coming from a protein in this particular case. Again, these rather large structures, and this particular one was discovered a little over 10 years ago, and unfortunately was lost. Getting back to your question about, can you keep these things in the lab? But more recently, one of my colleagues from Yellowstone showed me some images that look like this, only even more so. Just massive numbers of these bottle-shaped virions. So what's going on with that is not entirely clear. People argue about how they get inside the cell, maybe through the bottle end here, but maybe also through the filament ends, which are present right here. That's still a very open question. One of the big problems that people who are working on this had was it was very hard to get enough of these virions to study them in any detail. Usually the numbers of virions produced was very, very small. But now with these Yellowstone isolates, we hope that we'll be able to approach doing some of the analyses of these particular virions. There are also even newer ones. These are virions from what's called pyrobaculum. Pyrobaculum is another Archaea, grows at high temperatures, but at neutral pHs. These particular genomes are also linear double-stranded DNA with inverted terminal repeat structures. What's really interesting is that in these genomes, almost all of the open reading frames are all on one strand. And so it looks as if just one strand is being used, probably just one promoter is being used to make one messenger RNA, as opposed to both strands being used in terms of making all of the other proteins. Both strands are being used in all of the other viruses that we've talked about so far, certainly for the Fuselloviruses, the Lipotrix viruses, the Acidianus bottle-shaped viruses. So this one's interesting because it's just being using that one particular strand to make these. Also the virions, to me, this looks a lot like a enveloped virus that we see infecting a lot of eukaryotic cells. No obvious icosahedral symmetry, basically nothing is involved there. That being said, most of the virions that you see in some of these extreme environments do have these sort of spindle-like morphologies. But there are two classes of these spindle-like morphologies. Those like SSV, which have a short tail at one end. And then, when we were looking in the electron microscope, we saw all of these particles that looked like they had two tails, one on either end of the virion. And we had a really hard time isolating these. We found them in Yellowstone, actually in the late 1990s. Never able to isolate a host they could infect. David Prangishvili's lab, and I'll show you a picture of David right at the end here. Again, looking at Acidianus, this close relative of Sulfolobus. What they found was that they also had all of these virus particles with two tails on them. But then when they looked at infected cells, they saw that they were producing virions that looked like this. So, okay, what the heck's going on here? So eventually what they found out is that infected cells produced virions that looked a lot like SSVs. They had no tail whatsoever. But if you just incubated these virions at 70 degrees Celsius, they grew tails from either end of the genome. So there's a conformational change that happened in the protein that caused this maturation to take place. So we've talked about maturation happening outside the cell for what other kinds of viruses? The retroviruses, particularly for HIV, is the proteolysis that takes place. And that proteolysis is required for getting your final infectious nucleocapsid structure. Here, there doesn't seem to be any proteolysis required. It's just a conformational change. And what happens is you'll notice as these tails are growing down here at the bottom, the volume of this centerpiece is actually getting smaller. So the volume is being maintained. It's just the actual structure is getting different. So you have these short, fat, as it were, spindles or ovals here, which end up being long and skinny with two tails coming out at either end. The tails have these little hook structures at the end here, which is probably important for the infection process as well. These guys are non-infectious. These guys are infectious. So really fascinating, outside the envelope structure here being modified. I mentioned that most of these, in fact, these guys as well, have double-stranded DNA structures. This is one that was found about 4 years ago now by the colleagues who will be hosting me in Japan when I'm looking for new Fuselloviruses samples. Fellow by the name of Tomohiro Mizuuchi. He found this particular virus, but Trev had a question first.
Yeah, I'm just curious about the pH difference between inside the host as opposed to outside the host.
Okay, so the question is pH. And so what's the internal pH of these organisms versus the external pH? Acinianus is quite well known for being able to grow in media down to about pH 1.5 to 2. The internal pH is actually kind of hard to measure. I don't know if any has a pH electrode they can stick into the cell. But what you can do is you can get pH-sensitive dyes that will fluoresce at different pHs. And when you do these kinds of experiments, what you see is the internal pH of the cell is higher. It's usually about pH 5 to pH 6, but still relatively low, certainly relative to neutrality. Now, how the cells manage to keep an external pH and an internal pH so separate relative to each other, really open question. Maybe it's those ester lipids. Again, open question about how some of these things are able to do that. But it's a very interesting question. No one really has a good idea. These viruses themselves actually do seem to be stable at these low pHs. But again, what that really means, one of the things, we do leave these viruses at very low pHs for long periods of time. We do see a loss in infectivity. However, if we raise the pH a little bit, they seem to be fine for months. So there is some issue with the pH, and you get some DNA damage that happens there as well. Okay, so almost all double-stranded DNA viruses, with this one exception so far. This is Archaeal coil-shaped virus. Talk about funky-looking virions. It may be not quite as spectacular as the bottle-shaped virions, but it's this coil structure here with these two ends sticking out at either end. Who would have thought that this was a virion? It turns out not only is it a virion, but it's a virion which is packaging a single-stranded circular DNA. Now, how do you package a single-stranded circular DNA as this supercoiled structure? Again, we really don't know how it's being packaged here. Archaeal grows optimally at 90 degrees Celsius. This is the genome of that particular virus. You'll notice it's 25,000 bases, not 25,000 base pairs. This is about twice as big as the otherwise largest known single-stranded DNA virus found to date. Why you would have single-stranded DNA is anybody's question. Maybe it allows you to package in this form, but why should this form be particularly stable at these very high temperatures? Again, a really open question, and how you have packaging take place is a really, again, really open question. We do know something about packaging for these guys. What did I say about packaging for SSVs? The DNA is positively supercoiled, exactly. So there's another solution to this, and that's a solution taken by the Rudiviruses. So these are the stiff rod viruses, originally found in Iceland, Sulfolobus islandicus. Rod-shaped viruses 1 and 2. They're double-stranded DNA viruses. And what's interesting is the length of the virion is exactly correlated with the length of the genome. So if you have a bigger genome, you have a longer virion. You have a shorter genome, you have a shorter virion. And that tells you something immediately about the packaging. It's basically packaged as this helical structure, and packaged as this whole helical structure. The ends of the genome here were shown very nicely to be covalently closed hairpin ends. So just like what we saw with pox viruses, a lot of the other large DNA viruses, what's interesting here, which is different between those two, is that it turns out that these guys base pair all the way up to the end. And so instead of having a loop of unpaired bases at the very end of the genome, like we saw with the pox viruses, these could, at least in theory, base pair all the way to the end of the genome. So you can think of these as a linear double-stranded piece of DNA with covalently closed ends, or a single-stranded piece of DNA that's completely complementary. So just two different ways of thinking about the same thing, which is why I prefer monomolecular versus bimolecular. If you've got truly double-stranded DNA, it's 2 strands, whereas here it's 1 strand, which is just complementary to each other. So I prefer to think about it that way. These particular virions have extremely high mutation rates. So when you look at the changes that happen on replication in these genomes, they have almost as high mutation rates as RNA viruses do. What that means about the DNA polymerases, it seems to be host DNA polymerases which are replicating these. So why they have these high mutation rates is not clear at all. But it is known for other single-stranded DNA viruses that they also have pretty high mutation rates. I don't know if George talked about that at all when he gave his lecture. They're relatively small genomes, so you don't notice it as much. But in these genomes, you actually see that. These structures, as well as having these nice rods, which are, again, exactly the same length, or actually corresponding length to the genome, also have these tail fibers sticking out here at one end. Because these are such nice, well-behaved structures, you can do cryo-EM on these and then analyze in much more detail how the proteins are interacting with the DNA. And so this is a zoom-in cryo-EM published in Science last year of looking at the DNA together with these proteins. And if you look even closer to this, one of the things that people found is we have the structure of the capsid protein, but the DNA itself is A-form DNA. So we talked about A-form DNA, B-form DNA again way back when, the depths of molecular biology last term. These are very different double helical structures. So B-form, major groove, minor groove, similar kinds of sizes. The bases lined up really nicely relative to each other. Basically Watson and Crick form. You have to twist the DNA considerably. The base pairs don't line up nicely with each other, but it's actually a more compact structure. You've got a very deep major groove, a very shallow minor groove, and that seems to be stabilizing this particular genome. Again, Sulfolobus from these high temperature, low pH environments. These are non-enveloped viruses, so it's just protein and DNA, which is there, and that stabilization process seems to be happening again through this protein-DNA interaction. Yeah?
So is the coil virus, that we had 3 slides before,
Yeah.
different than the rod-shaped virus?
Yes, so it's a single-stranded DNA viruses versus a double-stranded, or at least completely complementary one. The sequences are not complementary, and the coil itself is much wider than this particular structure, which is a much narrower structure. So they're very different in terms of how they're packaged. We don't have anywhere near as high resolution structure for that coil virus as we do for these rod-shaped viruses.
Are the rod-shaped viruses similar to the plant rod-shaped viruses, except for
In terms of actual morphology, when you first look at them, yes. The plant rod-shaped viruses we talked about were which?
tobacco?
Tobacco mosaic virus. That's a RNA virus that actually does have a genome length and virion length that vary based on how long the genome is. And probably some of the packaging is similar, but it's a single strand rather than a double strand like we have here. So overall morphology similar, the details very different relative to each other. Okay, so I just wanted to finish up talking about a couple of fascinating aspects about these viruses. We already talked about the structure of STIV. I told you a little bit about this one, which we published in '99, but ended up, David Prangish really figured out quite what was going on there. There are also some of these giant viruses here, probably growing two tails as we're looking at them. This is the beautiful structure of STIV that we solve, but probably most exciting about this, other than the T=31, et cetera, icosahedral structure, is how these viruses actually get out of the cells. And this is a brand new virus exit mechanism that nobody had ever seen before. It was discovered in the study of, curiously enough, STIV, but also some of these rod-shaped viruses, Sulfolobus islandicus rod-shaped viruses. People were looking in the TM and noticed these very strange pyramid-like structures that they saw on infected cells that were either infected with STIV or SIRV. And then if you do thin sections where you cut through these, we have these pyramidal structures which then open up and make holes in the membrane. And this was shown even more detail a couple of years ago. David Prangishvili's group, they did electron tomography where you can see one of these pyramids before it opens, and then after it opens, it literally makes like this hole in the surface of the cell. And this is probably even more spectacular when you do a tomography of a whole cell producing these viruses. So here are a couple of, there's one of these that's just open, here's one that's completely open, and then here are some virions actually coming out of some of these holes that have formed, also known as virion-associated pyramids. People call these VAPs. But this is the SIRV, these rod-shaped viruses escaping. But it turns out that STIV, the icosahedral one, comes out in exactly the same way. They make very similar proteins to each other. Now, how do you end up with a icosahedral virus and a rod-shaped virus having similar proteins and similar exit mechanisms? Who knows? Again, interesting mystery, but clearly a lot of things to be discovered in studying these particular viruses. So I'll skip this, but basically saying, look, we have these really cool viruses in Archaea, the red ones here in the middle, boring eukaryotic ones we've talked about so far, and then the bacterial ones we talked about right at the beginning. This is just an overview, and this is the guy who infected most of this work after the two of us sat right next to each other in Wolfram Zillig's lab. He moved then to Regensburg, and he's now in Paris, and is doing almost all the rest of this work. It was his lab that discovered the bottle-shaped viruses, the viruses that grow the tails on the outside of the cell, the coil-shaped viruses, etc. So with that, it's 10:05, and if you want some more questions, come ask me. This is our sampling site in Lassen Volcanic National Park.